Thank you for the kind introduction, Dean Helbley. I don't know if that's a sign of my age that an intro can go on that long for me at this point.
Now's when you take questions.
So the topic of today's talk is really going to focus on my group's work on engineering enzymes as antibacterial agents. But maybe before we dive into the specifics of my research group's interests, I thought I'd take a minute just to make sure that we're kind of all on the same playing field as far as the subject matter and the types of molecules that we're going to be discussing in this engineering seminar today. All right, so my group does protein engineering, and proteins are in fact nanoscale machines. They have unparalleled activity and selectivity, specificity. These are unrivaled molecular machines with respect to the jobs that they perform. And the most obvious example of that is that it is proteins that are the machines of life. Proteins are what are enabling this discussion to happen right here today. They're enabling everything that you do on a daily basis. So proteins are really just heteropolymers of the 20 natural amino acids, by and large the 20 natural amino acids. So these amino acids are strung together end to end almost like Lego building blocks. So the amino acids get strung together into these long nanoscale strings, and they really are kind of molecular strings. And these strings then fold up into very compact globular structures. And these are the functional forms of proteins, right, that carry out the processes of life and that we're interested in as applied technologists. So I thought it would be useful to maybe remind you or introduce you to some of the ways in which applied protein technology actually impacts everyone in this room on a daily or at least very frequent basis, right? So outside of the biology of life, proteins, removed from their biological context, still maintain this exquisite activity that we can leverage to enable practical applications, all right? So a good example of how I imagine most of the people, or at least a good fraction of the people in this room, take advantage of applied protein technology. Who in the room is wearing clean underwear today? For those of you who aren't raising your hands, this is being recorded and will be posted on the web. So if you do laundry and you use any one of these laundry detergents or a number of other detergents to clean your clothes, you're taking advantage of applied protein technology. These detergents are formulated with enzymes, again, nanoscale machines that are designed to improve the cleaning power of these detergents. So they'll be formulated with lipases. These are enzymes that degrade grease-based stains in your clothes. They'll be supplemented with proteases that degrade protein-based stains in your clothes, for instance, maybe blood stains. They're formulated with a variety of carbohydrate hydrolases that can help remove stains, for example, from maybe grass stains in your jeans. All right, so again, these are nanoscale machines that are empowering better cleaning power within these detergents. I'll also make a quick comment on this slide about some of the renderings of these molecules that I'm going to be showing you today. So I'll be showing you a couple of different types of examples of protein structures. These are what are called ribbon structures, and these are intended to give you a feel for how these linear polymers fold up into compact structures. So you can actually see how the string folds up on itself into this complex structure. Again, this gives you a feel for what the peptide backbone of these proteins look like. I'll also be showing you images, some images that look more like this. These can be representations of the exact same proteins, right? But these might be molecular surfaces, and those molecular surfaces give you a better feel for what is the volume that is occupied by that protein. And I'll distinguish between these different representations when I show them to you. So we've got proteins in your laundry detergents. Likewise, we've got proteins supplementing the detergents that you may use to clean your dishes. So all of these dish detergents and numerous other ones are formulated with a couple of different types of carbohydrate hydrolases as well as proteases. This helps remove that stuck-on gooey food from your plates. Other examples of applied protein technology. If there's anyone in the room that drinks that, or if you enjoy a little bit of that from time to time, you've probably taken advantage of applied protein technology that goes into manufacturing these goods. If you wear contact lenses and you clean them like you're supposed to be, you're utilizing applied protein technology. There are enzymes to clean your lenses in those solutions. If there are people that like cheese, right? Oftentimes these cheese are actually manufactured with applied enzyme technology. If there's anyone in the room that printed out anything on paper at Thayer today, recycled paper is often manufactured with applied protein technology. If you filled up your car with gasoline that contains 10% ethanol, it's likely that ethanol was produced with applied protein technology, right? So this idea of protein solving practical real-world problems, it's out there right now. We're all taking advantage of it all the time. Now, in addition to consumer goods, applied protein technology has actually had an impact on the political system in the United States. So applied protein technology has gone into making a stronger political system. How do you take a scrawny kid like that, turn him into a hulking beast like this that can then become the governor of California? Number one, hard work. But hard work doesn't make you look like that. Right? Arnold Schwarzenegger took advantage of all kinds of performance-enhancing drugs during his career. It's possible. So the details of what he took are actually not disclosed, but it's possible that one of the compounds he might have used at some point would be human growth hormone. So this is a compound that can enable muscles to recover more quickly. It's used by elite athletes to enhance their performance. This human growth hormone has been at the center of a recent controversy on the U.S. Postal Service cycling team. Right? So our fallen hero, Lance Armstrong, and his compatriots, right? They have admitted to using human growth hormone to improve their performance in this sport. Now, in addition to human growth hormone being used for illicit purposes, it also has legitimate applications. In particular, this compound is used to treat children that are deficient in this growth hormone. They're not developing appropriately, and this compound can help treat that disease. And this really segues into one of my specific interests in the context of applied protein technology. How can we use proteins in order to treat human disease? And there are all kinds of examples of this that are out there. I suspect most people in the room know someone who's diabetic. Right? A lot of diabetics actually treat their diabetes by injecting insulin. That's an applied protein technology. In my family, my mother had a severe stroke in her 60s, but yet her life was extended for 7 years beyond that time, in part because the physicians were able to rapidly dissolve the blood clot that was causing that stroke, and they did that in part by using an enzyme called tissue plasminogen activator. Likewise, my father lived for years and years with cancer, and towards the end of his life was extended by a couple of years beyond what the physicians thought it might be. And in large part, that was because he was benefiting from applied protein technology. He was receiving an antibody that was designed to target and kill cancer cells. So this idea of using proteins to treat diseases is something that intrigues me, that fascinates me, and that's kind of very close to my heart, as well as, I think, very close to everyone's heart, right? This is something that really permeates society these days. And my group is focused on the application of protein engineering technologies to enable this type of therapeutic modality. Now, the stories that I'm going to tell you today are focused on one particular medical objective, and that is the problem or the specific challenge that a number of the projects in my lab are geared to address is the continuing and even accelerating spread of drug resistance among bacterial pathogens. So this is some graphics showing you trends of spread of antibiotic resistance among a few representative pathogens. So on the y-axis here, we're plotting the percent incidence of drug-resistant clinical isolates for 3 different bacteria. And we're looking at years from the early 1980s to the mid-2000s. And you can see in green here a general trend of increasing resistance for Staph aureus. Far more than 50% of Staph aureus are methicillin resistant now when isolated in the clinic. We have similar trends for an Enterococcus pathogen in red, as well as in blue, a bacteria called Pseudomonas aeruginosa, and this is going to be relevant to later portions of this talk. So again, there's this general trend of increasing drug resistance among these pathogenic bacteria. Now, at the same time, there's a frightening counter trend in the number of new antibiotics that are approved by the FDA. So we're looking at new antibiotics approved during 4-year time periods on the x-axis, and we're looking at from the early 1980s again to the mid-2000s, so that the same time frame here. And you can see this steady decline in the number of new antibiotics that are being approved. So we have these conflicting trends of increasing numbers of drug-resistant bacterial pathogens in the clinic and steadily decreasing numbers of new antibiotics that physicians have at their disposal to treat these types of infections. And this is really fueling a crisis that is being faced right now in the clinic, and this is a problem that's likely to get worse before it gets better. Now, what we want to do, our group, as well as other groups, are interested in the possibility of leveraging natural antibacterial proteins as a supplement to the physician's toolbox of drugs that are available to combat these severe bacterial infections. So my group is focused on engineering a number of different antibacterial enzymes. We have multiple projects in this intellectual space. But one of the enzymes that we've spent a lot of time focusing on is a natural antibiotic that's produced by my body and all of your bodies. This is the enzyme human lysozyme. So lysozyme is present in your tears, it's present in your saliva, in your nasal secretions, in your airway. It's produced by a variety of different immune cells that help protect your body. It's present in breast milk. So this is a widely distributed antibacterial enzyme in the human body. What I'm showing here up top is the primary amino acid sequence of human lysozyme. So the 20 natural amino acids, we give one-letter codes. Human lysozyme is composed of 130 amino acids that are strung together to make this long nanoscale string. All right, and that nanoscale string folds up into this compact three-dimensional structure that is the functional enzyme. So this is, again, a cartoon ribbon representation of the folded lysozyme structure. Lysozyme helps protect you from infection by attacking the bacterial cell wall. So bacteria frequently encounter environmental conditions where they're under enormous osmotic stress. And to protect themselves from bursting, they actually encase themselves in a gigantic molecular net called the cell wall or sacculus. So this is composed of a heteropolymer called peptidoglycan. And again, this net surrounds bacteria and it holds them together under enormous hydrostatic pressure. Now, what lysozyme does is it attacks particular chemical bonds in the cell wall. So it degrades, catalytically degrades the cell wall, eventually causing the bacteria to lose structural integrity, to lyse or pop, and that kills them. Now, I'll point out that the basic foundation or the fundamental structure of peptidoglycan is shared commonly across the vast majority of bacteria. So there are some subtle differences in peptidoglycan from one bacterial species to another, but in general its structure is conserved. And what that means is that lysozyme, under the right conditions, is a broad spectrum antibiotic. It can kill a variety of different bacterial species. I'll also point out that lysozyme is a catalytic antimicrobial agent, right? It can degrade many bonds, tens, hundreds, thousands, tens of thousands of bonds in a single bacterial cell. This is not a molecule that binds once and that's it. This is a biocatalyst. So just to give you a feel for how lysozyme attacks and kills bacteria, I've got a couple of videos here. So in this video over on the left-hand side, what I've got are a couple of bacterial suspensions. So I've got bacteria in solution in the same concentration in these two tubes and a clear buffer solution here. What I'm going to do is I'm going to inject a little bit of human lysozyme into this bacterial suspension, mix that up, and you're going to see that as the enzyme starts attacking those cell walls, it's disintegrating those cells, and you'll see this solution start to go progressively more clear. You can already tell the difference right there. Now, if you were to look at this on a microscopic scale, what you would see is there are individual bacteria in there that are being attacked by the enzyme. Their cell walls are being degraded, and they are just being dissolved, literally lysed. That's the same thing going on here, and eventually this solution would go clear, almost as clear as that solution, given enough time. So this gives you some perspective on how lysozyme, right? That's pretty good. So this gives you some perspective on how lysozyme attacks these bacteria, and it literally disintegrates their cell wall, causing them to burst and kill them. So the take-home message here is that lysozyme is actually a critical component of your innate immune system. It helps protect you against bacterial invaders, and it is, under the right circumstances, a very powerful antibacterial agent. Now, the problem with lysozyme is that under some disease conditions, it's not operating at its optimum efficiency or efficacy. So the two stories that I'm going to tell you today deal with projects where we've identified conditions that cause lysozyme to operate under less than optimal conditions, where lysozyme is inhibited non-specifically by just the harsh environmental conditions of an infection. And then likewise, a second project where we're looking at specific lysozyme inhibitors that are produced by pathogens to actually protect themselves from your immune system, and how we might be able to address that. So these are the two stories that I'll tell you about today. So this first one, this non-specific inhibition of lysozyme due to the infection environment, this ties into my research group's interest in cystic fibrosis. So there's actually an outstanding group of researchers here that study various aspects of cystic fibrosis, a very powerful research team here at Dartmouth. Cystic fibrosis is a genetic disorder, right? It's caused by mutations in a gene that encodes for an ion transporter. So this protein, this ion transporter, doesn't work right in CF patients. And there's a number of clinical complications that are associated with that. But perhaps the most damaging is the fact that these patients become highly susceptible to lung infections. So this Pseudomonas aeruginosa bacteria that I mentioned is one of the emerging drug-resistant strains that is out there. This bacteria can colonize these patients' lungs. When it infects their lungs, it's very difficult to eradicate. It's difficult to eradicate with antibiotics. The patient's immune system cannot effectively clear these bacteria, and it leads to really kind of a vicious cycle of chronic infection and inflammation in the lung. And what happens is that the lung tissue eventually starts to become progressively degraded. So here's a chest x-ray of a CF patient, and you can see scarring of the lung tissue as kind of these white clouds here. Compare that to a chest x-ray of a healthy individual. All right, so in this infected lung environment, lysozyme is not participating effectively in clearing these bacterial pathogens. And part of the reason for that, so lysozyme is very prevalent in your lungs, but it's not clearing these infections. Part of the reason for that is that in the infected CF lung, there is a dramatic buildup of negatively charged biopolymers. So for instance, DNA is the biopolymer that actually encodes, that's the genetic blueprint for our proteins. There are a number of different types of cells that are being lysed within the infected lung environment, and they are spilling their DNA out into that lung space. So DNA can build up to high concentrations in the infected lung. DNA is densely negatively charged. F-actin is a structural protein that contributes to the structure of cells. Again, this is a densely negatively charged protein that is spilled into the lung environment during these severe lung infections. Mucin is a glycoprotein that's naturally present in our lung tissues. Again, this is a densely negatively charged glycoprotein, and upon infection or upon a bacterial invading the lung space, mucin concentrations can skyrocket really rapidly. And then finally, Pseudomonas aeruginosa itself produces a negatively charged biopolymer, a negatively charged polymer called, or carbohydrate called alginate. Right, so the point here is that in the infected CF lung, all of these densely negatively charged biopolymers build up to relatively high concentrations. And this is bad for lysozyme because it turns out that lysozyme is densely positively charged. So what I'm going to show you here is an alternative representation of the human lysozyme molecule. As opposed to the ribbon structure that shows you how that string folds up, this is actually a map of lysozyme's molecular surface. So this is the volume that lysozyme kind of carves out in space. Again, this is really just about a 3 nanometer protein here. So what we're looking at is the molecular surface of lysozyme, and I've mapped onto this surface the electrostatic potential of the protein. So positive potential is in blue, negative potential is in red. And what you're going to see as this molecule is rotated in 3D space is that the vast majority of this molecule has cationic character. It's positively charged. There are just a few spots on this protein that have this negative electrostatic character. The primary spot is right here in the enzyme's active site. This is actually the active site cleft, so that's kind kind of the edge of the scissors that lysozyme uses to cut those bonds in the bacterial cell wall. But for the most part, lysozyme is densely positively charged. So in the infected lung, we have a buildup of negatively charged biopolymers like DNA and alginate, mucin, and F-actin. We have antibacterial lysozyme, which is densely positively charged. And not surprisingly, there are coulombic attractive forces between these oppositely charged species. And lysozyme is subject to electrostatic-mediated aggregation and inhibition by these polymers. So these are some photographs showing you that a lysozyme solution by itself is clear. Lysozyme's totally soluble. An alginate solution with no lysozyme is completely clear, but you mix those two solutions together and you get an immediate precipitate formation. A similar effect with mucin. Mucin without lysozyme, relatively clear. You add lysozyme and you get copious precipitate formation, and a similar effect with DNA. You mix a little bit of lysozyme with DNA and you get kind of these white tendrils of precipitate that form rapidly. All right, so the idea here is that lysozyme, wild-type human lysozyme, is subject to electrostatic-driven aggregation and inactivation by these biopolymers that accumulate in the infected lung space. So the objective of this project is to redesign human lysozyme so that it exhibits improved antibacterial activity in the infected lung. And by way of giving credit to the individuals that actually did this work, I'll note that Dr. Thomas Scanlon, a research scientist in my lab, this is really his project, his baby. He did the majority of the work that I'm going to show you here. He had a little bit of assistance on some structural analysis from a postdoc, Dr. Avinash Gill in the lab, a former postdoc. And then we have a collaborator, a physician scientist at the University of Vermont School of Medicine that helps us conduct some in vivo efficacy tests in a model system. All right, so the idea here is we want to engineer lysozyme to evade electrostatic-mediated aggregation and inhibition in the infected lung. How are we going to do that? Well, what I'm showing you here, again, is the primary amino acid sequence of lysozyme. So there's 130 amino acid residues. It turns out some amino acids are positively charged. And what I've done is I've highlighted all of the residues in blue are positively charged amino acids. Each of these amino acids can carry a charge of +1. So in aggregate, these amino acids contribute to the net cationic character of human lysozyme. And if you want to reduce its electrostatic attraction to these negatively charged biopolymers in the lung, well, it's a pretty intuitive thing to think, well, let's just change these positively charged amino acids. And we can use genetic techniques to rapidly switch out any one of those amino acids that we wanted to. So we could mutate any of these positively charged residues to a neutral amino acid or even a negatively charged amino acid, reduce lysozyme's net charge, and probably reduce its propensity to be attracted to these negatively charged biopolymers. What I'm going to show you here is a quick movie just showing the mapping of all of these positively charged residues across the surface of lysozyme. So you'll see some spots where there's several residues that are kind of spaced together closely, but in general these residues are spread out, kind of distributed across lysozyme's surface. So it'd be easy to mutate these and reduce net charge, but you've got to remember that our ultimate objective is not simply reducing lysozyme's attraction to these negatively charged biopolymers. What we want that enzyme to do is kill bacteria. And a critical component to how lysozyme homes in on bacteria and attacks bacteria is, in fact, some portion of these positively charged residues. So if you mutate the wrong residue, you'll kill its antibacterial activity. And this is a non-intuitive protein design problem. You cannot look at this sequence or look at this structure and say, oh, I need to mutate that lysine to an alanine, and that's going to solve the problem. Right? That's not something that we can do or achieve. So instead, my research group oftentimes leverages what I refer to as combinatorial protein engineering techniques. And the reason for that is, again, because this idea of rational design, rationally picking out the mutations to make, is just simply intractable in this case. So the idea of combinatorial engineering, maybe just as a quick reminder of the central dogma of biology, right? We are protein engineers. We want to engineer these functional biomolecules that I'm representing by this blue shape here. But we're not going to do that by directly manipulating proteins themselves. That's actually a non-trivial thing to do. It's much easier to change the composition of a protein by mutating the DNA that encodes for that protein. So we can mutate the double-stranded DNA, the gene that encodes for this protein, and thereby change the protein structure and change the protein function. So our combinatorial approach to protein engineering relies on the fact that we're not going to make one rationally informed mutation to this lysozyme gene and hope that that solves our problem. We're going to make lots of different mutations to this gene. So we're actually going to build a library of mutated lysozyme genes. And we may build thousands, tens of thousands, hundreds of thousands, in some cases millions, tens of millions, billions of different genes. And the idea here is that if we build a sufficiently large repertoire of mutated lysozyme genes, and they're of sufficient quality, well then it's likely that somewhere within this repertoire of mutated genes, there's at least one mutant gene that encodes for a lysozyme variant that has the improved properties that we're interested in. Specifically for this project, again, we wanted to kill bacteria in the presence of these negatively charged biopolymers. So how do we build these mutant libraries? In this particular project, Tom Scanlon looked at these 20 positively charged residues, and he actually did a bioinformatics analysis, and he did some structural analysis, and Tom picked out 8 of these positively charged residues that he wanted to target. And the reason for this is that we needed to build a reasonably sized library. If we were going to do combinatorial mutagenesis on all 20 of these amino acids, the library would be far too big for us to effectively analyze and screen. So Tom focused it down to these 8 residues, and then what he did is he mutated these wild-type histidine or arginine, predominantly arginine residues, to one of 3 alternatives. So he would mutate each one combinatorially to an asparagine, which is a neutral polar residue. So this would, each mutation to an asparagine would reduce the net charge by 1. Alternatively, he mutated to an alanine. This is a different amino acid that is likewise neutral, but it's nonpolar, so its chemical character is a little bit different than it is here. And then finally, as a third alternative, he mutated them to aspartic acid. So these are negatively charged amino acids that would reduce the net charge by 2 for each position where we made this substitution. And again, the idea is that so if we have 8 target sites and 4 possible amino acids that can be filtered in at each site, then we have a theoretical library size of 65,536 different mutant genes that encode for 65,536 different protein variants. So that's a lot. That is a lot of proteins if you're thinking about testing these proteins. But this actually is a relatively manageable library size. So this is the strategy by which this library was constructed. Details are in this 2009 publication. Now, the second part to combinatorial protein engineering, once we build the mutant DNA library, right, we're interested in the proteins. We want to engineer the proteins, but we're doing that through the DNA. So once we have this mutated DNA library, we want to use that to build proteins, to build the mutant proteins. And we need to do this in a carefully designed fashion such that we maintain a physical link between the mutant gene and the protein that it encodes. And the reason for maintaining this tight coupling of genotype and phenotype will become apparent in just a minute. All right? But it's important that we do this. Now, how we do it, how we accomplish this in my lab, is typically we compartmentalize the genes and the proteins inside microorganisms, recombinant microorganisms. So again, this is a mutant lysozyme library. This might be the wild-type lysozyme gene is the white block. Each one of these colored bars in the other blocks represents different mutations to the human lysozyme gene. So we build a combinatorial library of mutant lysozyme genes. We clone that into circular pieces of DNA called plasmids that are able to replicate in these microorganisms. And then we can transform these plasmids into a recombinant microbial expression host. So this is a cartoon schematic of a yeast. These yeast cells might be 4 or 5 microns in diameter, and the biology of yeast is such that ultimately each yeast is going to be producing one and only one type of mutated lysozyme from our original library. All right, so we've compartmentalized the mutant genes, and the yeast are producing the lysozyme encoded by that gene and secreting it to the environment that's close to that yeast cell. Now, to separate these yeast cells, because when we make these libraries, we can actually do this pretty effectively and pretty quickly, but we form these libraries in a single tube. So in order to separate all these yeast clones so we can look at each one individually, what we do in this case is simply pour the yeast onto nutrient agar media and spread those yeast out to single cell resolution. So we spatially separate each individual yeast, and because they're sitting on top of nutrient media, they start to grow and divide, and eventually they go through enough divisions that there are a sufficient number of yeast that you can actually see the colony of yeast. So inside this dot, right, we have many, many yeast, and they're all exact genetic clones of each other. So all of the yeast in this particular white dot are producing exactly the same type of lysozyme, and it's only one of the lysozymes that was encoded in our original library. So this is the idea of tying genotype to phenotype. We actually use biology to do that. Now, the final piece of the puzzle in combinatorial protein engineering is once we have our mutant gene library, we've converted that mutant gene library into a mutated protein library where these proteins are related to our wild-type protein, but they're all subtly different. The objective is to go through and screen each one of these proteins and test it for the activity of interest. So we have to develop high-throughput assays that allow us to assess whether or not these mutated lysozymes are killing bacteria in the presence of the polyanion inhibitors. So if we can identify even one of these mutated proteins that is accomplishing that goal, then we can actually specifically find out what the mutations are in this protein that enables it to kill bacteria in the presence of the polyanion inhibitor, and we can do that by sequencing the gene. So by tying the original gene to the protein with these yeast cells, again, we maintain this genotype-phenotype link, and because we have the mutant gene, we can now use recombinant DNA technology to produce lots and lots of this protein of interest. So how are we going to go about assaying for our desired activity? In this case, Tom used a plate-based screening assay that's called a halo formation assay. So he simply formulated yeast growth media. That's kind of this yellow cylinder here, right? This is yeast growth media. You put yeast on top of this media, and they start growing and dividing, and they form these colonies here. But he also embedded within this media reporter bacteria. So these blue dots represent bacteria that we would like to attack and kill with our lysozyme. Now, the idea is that if one or more of these yeast are secreting an active lysozyme into this media, it will diffuse through the media, attack those bacteria, lyse them, and we'll get a zone of clearance, right? Or actually a halo forming around that bacteria. So this is a photograph of one of these indicating plates, and what you can see here are two yeast colonies. Both of these yeast colonies are producing wild-type human lysozyme. You can see that there are these zones of clearance around the yeast. These are really halos where the lysozyme that's produced by these yeast clones is diffusing into the media and attacking and killing the bacteria, and allows us to easily distinguish these yeast producing an active enzyme from yeast producing a non-active polypeptide. Now, this is in the absence of any of those negatively charged biopolymers from the CF lung. What if we include one of those biopolymers in this media? So when Tom formulates this media, in this case with 2% alginate, this is one of the biopolymers in the CF lung, you can see that now suddenly the yeast producing wild-type human lysozyme, they have no zone of clearance. The reason for that is that the alginate is electrostatically attracting and inactivating that wild-type lysozyme that's being secreted. So now, right, you can look forward to how we're going to go about screening these libraries. We have a library of different yeast clones, each yeast producing one and only one type of charge-engineered lysozyme, and we can spread this mixed population of yeast on indicating inhibitory media, and we'll look for those individual clones that are generating halos. These are the yeast that have genes encoding enzymes that escape this electrostatic-mediated inactivation. So Tom actually screened hundreds of thousands of yeast looking for these types of halos, and I got to tell you, this image, this is definitely enhanced. Identifying these zones of clearance is not nearly as easy as you might think it is. So it was an enormous amount of work, but ultimately he actually found a pretty large number of engineered lysozymes that were forming these halos on inhibitory media. So we picked out one of these enzymes that was of particular interest, and we purified it and carried out some detailed characterization in the lab. So one of the things that we're interested in is how fast does this enzyme attack and kill bacteria? And I showed you earlier that video, right, where we added a little bit of lysozyme, and you could see this solution going clear with time. Well, we can quantitatively track the number of bacteria that are present in this solution as a function of time, and then do kinetic analysis on how quickly is an enzyme degrading the bacteria. So in the absence of any inhibitor, this is a bar graph just giving you one of the readouts of relative catalytic efficiency for the wild-type enzyme in blue and Tom's most promising charge-engineered variant in red. And so by this measure of activity, the engineered variant is actually about twofold more efficient than the wild-type enzyme. And this is in the absence of inhibitor. What we're really interested in is what happens if we start putting alginate or DNA or mucin into that media. How fast do these enzymes lyse bacteria with these inhibitors around? So this is a bar graph where I'm just plotting kind of inhibition susceptibility. So this is relative susceptibility to inhibition on the y-axis, and it's inhibition by alginate, mucin, or DNA. So the higher these bars, the more susceptible this enzyme is to inhibition by this biopolymer. And again, we've got wild type in blue, our engineered variant in red, and the take-home message is that the engineered variant is about threefold less susceptible to inhibition by alginate, about sevenfold less susceptible to inhibition by mucin, and more than 40-fold less susceptible to inhibition by DNA. Right, so this is exactly what we wanted this enzyme to do. In the laboratory, it's killing bacteria in the presence of these negatively charged biopolymers. But ultimately, we'd like it to kill bacteria in the infected lung environment. So we work with a collaborator who has a mouse model of lung infection by Pseudomonas bacteria. So Lori can infect the lungs of mice with a clinical isolate of Pseudomonas bacteria. 1 hour later, she treats those mice with wild-type lysozyme, Tom's charge-engineered variant, or a sham treatment, so just a blank buffer treatment. And then the next day, she has various readouts of efficacy. How effective was this enzyme at combating that lung infection? So the data that I'm going to show you here is just data from one experiment, and we're really just looking at CFUs per lung. So this is the number of live bacteria that were left in the lung 24 hours later. So here's the sham treatment with no therapeutic agent in there whatsoever, and you see that the median is about 1.8 million bacteria per, or on average, these mice have 1.8 million bacteria in their lungs. If those mice are treated with wild-type human lysozyme, we get a substantial reduction. So only about 500,000 bacteria in the lungs of mice that are treated with wild-type human lysozyme. You treat them with the charge-engineered variant, and in this case, there's a pretty strong trend towards even better efficacy. So in this case, the mean was in between 100 and 200,000 bacteria per lung. So you can see that we can achieve a dramatic reduction in bacterial burden by treating these mice with this charge-engineered variant. So how does this enzyme accomplish this improved activity? Why does it work better? It's because we've redesigned the electrostatic potential. So this enzyme was actually crystallized, and what we're showing here again is the polypeptide backbone. So the way that that molecular string folds up is shown in yellow for wild-type human lysozyme, and then the fold for the engineered lysozyme is shown in yellow over here. So it's tough to see in this graphic, but those folds, the global folds of these two enzymes, they're essentially identical. There's very little difference in the global fold of the enzymes. But what I'm showing here is actually a projection of the electrostatic potential field that emanates from these proteins. So again, we've got positive potential in blue, negative potential in red, and you can see the wild-type enzyme has this huge, massive, positive electrostatic potential field that it projects into the surrounding environment, whereas the charge-engineered variant, it has a contracted electrostatic potential field, and it also has expanded area of negative potential. So it's this altered or reshaped electrostatic potential field that enables this enzyme to escape inactivation by negatively charged biopolymers, but at the same time still maintain high-level antibacterial activity. All right, so this is really kind of where this project has wrapped up for the time being. And the summary key points is that we're interested in biocatalysts as next-generation antibiotics. In the case of human lysozyme, It is subject to electrostatic-mediated aggregation and inactivation in the infected lung environment, and we've used combinatorial protein engineering to design new lysozymes that show improved activity in this infected lung environment. So this is the story of lysozyme being inhibited kind of non-specifically by the harsh environment of a lung infection. So in the last few minutes here, I want to tell you about an ongoing story in the lab where we're looking at inhibitors of lysozyme that are produced by bacteria. So some bacterial pathogens have actually evolved proteins that they produce in order to specifically protect themselves from destruction by lysozyme. So this is a 2007 PNAS paper where they did a structural as well as a phylogenetic analysis of a particular class of these lysozyme inhibitors. These are the inhibitor of vertebrate lysozyme, or the IV proteins. Right? So this is a class of proteins that was found in gram-negative bacteria just in the last decade or so. And a year after this publication, a collaborating group actually showed that these IV proteins helped bacteria survive in fluids that had high concentrations of lysozyme. So for instance, E. coli can grow in hen egg white, in human saliva, and in human breast milk. It can survive in these biological fluids that have lots of lysozyme, specifically because it produces this IV inhibitor that protects this bacteria from killing by lysozyme. All right, so the problem here is that bacteria can specifically shut down lysozyme's antibacterial activity. So our objective in this project is to redesign human lysozyme to evade inhibition by these pathogen-derived proteins. And the folks who spearheaded this work would be Dr. Sarah Dostal, a postdoc in the group who did the library construction and screening. Tom Scanlon, again, actually developed and optimized the high-throughput screen that I'm going to show you here. And then a couple of graduate students, Youngling Feng and John Garrett, are now collecting data on some of the enzymes that we've isolated. So again, the idea here is that we want to design an enzyme that will evade inactivation by the IV protein. So once again, I'm rendering here a molecular surface. So this is the molecular surface of human lysozyme. There's that active site cleft I've shown you before. I'm going to rotate this thing by 90 degrees, and then I'm going to show you how the IV protein, so this is human lysozyme in gray, this is actually an IV protein in blue. And this is in very tight complex with this human lysozyme. It kind of binds rigidly to this interface of the lysozyme protein. And if we turn this so that we're looking at it from the top down, here's the active site cleft of lysozyme that it wants to use as scissors to degrade the cell wall. And you'll notice that IV actually projects an amino acid residue right into that active site. So it occludes the active site and shuts down lysozyme's antibacterial activity. So the idea here is that we wanted to identify which residues in lysozyme were responsible for this interaction. So what I'm going to do is separate these two and then show you the binding face of lysozyme. So this is the binding interface where IV complexes with human lysozyme, and it inserts those residues right into this active site. So what we did is we specifically identified a number of amino acid residues on lysozyme that are responsible for close contact with the IV protein. All right, so we identified 6 amino acid residues. Now in this case, we don't simply want to change charge. In fact, some of these target sites are not even charged. What we want to do is we want to change the shape. Right? We want to change the chemical characteristics of these side chains and see if we can disrupt this IV lysozyme interaction. And again, this is a problem where there's no way to tell a priori which substitutions will do that while also maintaining high levels of antibacterial activity. So we once again use combinatorial protein engineering. We take these 6 sites and we combinatorially substitute in 12 different natural amino acids. So we get a library that's pretty close to about 3 million different members. Right? This is a much bigger library than the last library that we were talking about. Right? So significantly larger than the charge engineered library. And what that means is that when it comes to this issue of designing a high throughput screen to test for the lysozyme activity of interest, we need a more efficient assay. This plate-based halo formation assay is not going to cut it in this case. So I've already shown you that we utilize recombinant yeast, right, to compartmentalize our mutant genes and to convert each of these mutant genes to the corresponding engineered protein variant. So what Tom has done here is he's actually developed what he calls a gel microdroplet assay. So he's actually going to compartmentalize, again, each one of these yeast in a picoliter volume reaction flask. And he's going to do that through microfluidics technology. All right, so this is a schematic of a cartoon schematic of what the microfluidics might look like. And I just want to focus on that for a minute because it's an important point. So this is one of the chips, or similar to one of the chips that he's looked at. This is a close-up of the microfluidics channel here, and again, the cartoon schematic. So the scale that we're looking at here is that each of these little droplets is maybe 30 to 50 microns in diameter. And the way that this works is that we have an aqueous phase that's flowing onto the chip, and then at right angles we have an immiscible oil phase. And due to the fluid dynamics here at this junction, this aqueous phase breaks off into discrete individual droplets of controllable size. And the way that this aqueous phase is formulated is that there's kind of three key components that we put in there. So one, we put in our lysozyme library, where each yeast is producing one and only one of our mutated lysozyme variants. We also put in lots of different, or a large number of target bacteria. So that's these red dots here indicate bacteria that we would like to kill with lysozyme in the presence of the IV inhibitor. And the last thing that Tom includes here is a gelling agent, all right, so that we can actually convert this aqueous solution into a solid hydrogel, or a solid hydrogel matrix. So the idea is that this is formulated such that on average, each droplet has no more than one of our yeast from our original library, but each droplet has many, many target bacteria. So we've encapsulated a large number of target bacteria with a single yeast clone. And the idea is that we can flow these gel microdroplets, or these inverted emulsions, off the chip. So we have a continuous oil phase. Dispersed throughout it are these aqueous droplets. We can change the conditions so that these droplets actually gel and form this solid hydrogel matrix that, again, is now encasing the target bacteria with the yeast, producing our lysozyme. And once we gel these droplets, we can break the inverted emulsion and resuspend the droplets in a continuous aqueous phase. And now what we can do is we can actually take purified IV protein. So the IV protein inhibits wild-type lysozyme. We can put that purified IV protein in this aqueous phase. It will diffuse into these droplets, and any enzyme that's like the wild-type enzyme, its activity will be shut down, and those gel microdroplets will have live bacteria, so the target bacteria will survive. Whereas if we have a few microdroplets with yeast secreting an active human lysozyme that evades the IV inhibitor, it will attack and kill these target bacteria, and we can actually stain these dead bacteria using a fluorescent molecule. So this is a probe that will fluorescently label only dead bacteria. It won't touch live bacteria. And the idea now is that based on this differential fluorescent signal, we want to physically separate these two GMD populations. So these are yeast that are producing lysozymes that evade the IV inhibitor. Now, how we isolate these fluorescent gel microdroplets is using flow cytometry technology. All right, so the general idea here is that flow cytometers, we can take a mixed population of, say, GMDs in this case. And again, only a few of these GMDs have lysozymes that will kill bacteria in the presence of IV, and only a few of those will thereby be stained with that fluorescent probe. Most of them are non-fluorescent. We flow this mixed GMD population at a high rate of speed through an intersecting laser beam, and based on the intersection of that laser with the GMDs, we can collect a variety of spectral information about these gel microdroplets. Maybe most importantly, we can specifically identify those gel microdroplets that are fluorescent. These are the GMDs that have dead bacteria in them. And then the power of flow cytometers is that we can actually physically separate the non-fluorescent GMDs from the fluorescent GMDs. So based on their spectral characteristics, we can physically separate these two types of gel microdroplets. The instrument that we have at Thayer, right now we're doing this at a rate of about 3,000 gel microdroplets per second. Right? That's a phenomenal throughput. This instrument actually has the capability to go much, much faster. But you can imagine that with this throughput, we can rapidly go through huge libraries of lysozymes looking for those that are able to kill the bacteria that are co-encapsulated in that gel microdroplet. So I think that in the interest of time, what I'm going to do is maybe show you what these gel microdroplets look like and then just skip to the punchline. So these are some fluorescent micrographs of gel microdroplets. And what I've done here is I've outlined the gel microdroplet with a dotted white line because it's actually hard to see its outline. The white arrows here point to yeast colonies. So these are yeast that are secreting wild-type human lysozyme in this case. Now, peripheral to the yeast colony, you see these other clumps of cells. These are actually the target bacteria. So a yeast secreting wild-type human lysozyme in the absence of IV kills those bacteria, and we get a bright fluorescent signal in that GMD. If we throw that GMD, where yeast are secreting wild-type human lysozyme, into solution with IV, we completely shut down the fluorescence intensity, or we shut it down almost completely by virtue of inhibition of that secreted enzyme. So we can, again, use flow cytometry to separate these two GMD populations based on the fluorescence characteristics. So we've screened using flow cytometry millions of these GMDs, and we've actually isolated some lysozymes that are able to evade IV inhibition. So what I want to do is just show you some of the data that's hot off the presses. This was actually generated last week. All right, so what we're plotting here is another kinetic experiment. So we're monitoring the rate at which a lysozyme degrades and lyses bacteria in solution here. So we measure this rate in the absence of the IV inhibitor. That's this rate naught. We then add a little bit of IV into a replicate reaction, and we measure the rate of the enzyme in the presence of a little bit of IV. And this is the rate of degradation in the presence of IV. And we can do this for a range of IV concentrations. And so if we plot this ratio of the rate of degradation of bacteria in the presence of IV divided by the rate in the absence of IV, on the y-axis, we kind of get this proportion of uninhibited activity that's maintained at any given IV concentration. We can actually fit this with an appropriate model and extract out apparent affinity constants. So for wild-type human lysozyme, this IV protein has an apparent dissociation constant of about 12 nanomolar. That's a really tight binder. All right, and you can get a feel for that by looking at this. At 100 nanomolar IV, we've reduced the lytic activity of wild-type human lysozyme down to less than 10%. If we go out here to 400 nanomolar IV, we have very little detectable lytic activity for wild-type human lysozyme. Two of the enzymes that were isolated with this FACS GMD assay are the engineered variant G11 and the engineered variant 2A1. So these are plots that I'm showing here, or analogous plots to what we showed for wild-type human lysozyme. Notice the difference in scale. This goes out to 500 nanomolar IV as opposed to 500 nanomolar IV. So if we come back here to 400 nanomolar IV concentration, where wild-type human lysozyme is virtually completely shut down, G11 maintains about 25% of its lytic activity. In the case of 2A1, it maintains more than 70% of its lytic activity at 400 nanomolar. So these are enzymes that are able to evade molecular recognition by IV and yet maintain an inherent capacity to attack and kill bacteria. We're in the presence of characterizing these enzymes further right now. So the summary here is that we have developed a powerful high-throughput screening assay for antibacterial proteins. And you can imagine that we could apply this in lots of different ways. What we've applied it to so far is engineering human lysozyme, which is susceptible to inhibition by proteins produced by pathogenic bacteria. In particular, the IV protein is what we've looked at. Once again, we used combinatorial protein engineering to develop lysozymes that evade inactivation by the IV protein. And our long-term goal is that we would like for some of these enzymes, or at least the basis and the technology for some of these enzymes, to perhaps be candidates for translation into therapeutic agents, right, for treating drug-resistant bacterial infections. So in general, one of the main focuses of the group is engineering biocatalysts as next-generation antibiotics. So the last thing I'll do is give credit to the rest of the group. So I showed you the people that were responsible for the work that I showed you today. My group is much larger than that. We have a variety of different projects, so I'll acknowledge the top-notch group that I have here at Thayer. I have a number of really important collaborations. Lori Whitaker at the University of Vermont, who I talked about today. Chris Bailey-Kellogg over in the computer science department. We have a phenomenal collaboration on a different topic than what I talked about today, and a new collaboration with the professor over in the Geisel School of Medicine. We have funding from various national institutes as well as private foundations. I should also acknowledge the phenomenal support that I've gotten from the Thayer School and the administration of the Thayer School. So, right, this financial support makes this research possible. And with that, I thank all of you for your time today. Yeah. That's
what makes the lysozyme not attack humans?
So,
and a related question is, this is sort of genetic engineering of human proteins, is it safe?
So, in answer to question 1, lysozyme is exquisitely specific for bacteria because humans actually don't have peptidoglycan cell walls. We have no structure like peptidoglycan, and lysozyme specifically degrades that peptidoglycan. So lysozyme is essentially inert to human cells. Genetic engineering of human proteins, right? So there are certainly concerns that could arise. For instance, if we create a variation of lysozyme and administer that to a patient, it's possible that the patient's immune system might recognize the subtle changes that we make and say, wait a minute, that's a foreign agent, and actually mount a response against that, attack that therapeutic agent. And the real risk is, well, does the immune response against our engineered lysozyme also attack the wild-type human lysozyme and thereby degrade your inherent antibacterial protection or protective mechanism? So this is certainly a concern, and this is actually the focus of the collaboration that I have with the Dartmouth Computer Science program, is we are looking at what drives immunogenicity in proteins. So that's something that we're aware of and is part of the long-term plan for these agents.
Is there any possibility of this sort of getting out, the body saying, I like this, I'm going to move over to what I've generated and start generating this stuff?
So in the case, what I'm talking about here is I'm actually talking about proteins that we're using as therapeutic agents. So we produce these in recombinant microorganisms, but then we purify the protein. So the DNA, the genetic information is gone. It's just the protein agent. And so there's no potential for replication there.
Is lysozyme generic? Namely, is mine the same as yours?
By and large, yes. There are actually some diseases that are associated with mutations to lysozyme. So lysozyme can actually form plaques. And that causes all kinds of clinical complications. But for the most part, your and my lysozyme are the same molecule. Yeah?
So these proteins are injected into a patient and bacteria killed. Have you looked at, or has anybody looked at, like how fast they adapt to the new lysozyme, or how fast they can re-engineer themselves to produce a new item?
Yeah, that's a great question. All right, and this is actually one of the key limitations of the antibiotics that are used right now, is that bacteria adapt to small molecule inhibitory antibiotics lickety-split. So I've got statistics in another slide that show that for some antibiotics that are on the market right now, resistance was detected the same year the FDA approved the antibiotic. So bacteria have the ability to rapidly evolve resistance to antibiotics. Our interest in biocatalysts, and in particular biocatalysts that attack the cell wall or the cell membrane, is that there is evidence that these types of antibiotics, the antibiotics that your and my body has engineered through evolutionary processes, that they've been engineered specifically to try to avoid the development of rapid resistance. So the bacterial cell wall and the bacterial membrane are not directly encoded in genetic information. They're the result of large, complex synthetic and transport pathways that involve multiple enzymes in the assembly and transport of these constituents. So if you're going to mutate the cell wall of a bacteria to evade killing by a lysozyme that normally would attack that bacteria, you have to mutate potentially multiple genes, multiple enzymes in that biosynthetic pathway, and they have to still remain integrated and functional. So it's a more difficult evolutionary barrier to develop resistance against agents that attack the cell wall or the cell membrane. And again, so there's evidence of this in the literature. This is actually an area that we're interested in exploring. Maybe one more question? Absolutely, right? So that in this way, right, the bacteria could potentially respond rapidly by developing an IV that can actually complex with that lysozyme. So the long-term goal might be, or one potential solution might be, you actually develop a cocktail of lysozymes, right, where there's different molecular substitutions that have been made to avoid that IV. So if the IV adapts to bind one of the new engineered lysozymes, one of the other ones can fill in the gap, and thereby you wipe that bacteria from the population. Yeah, maybe one more?
On stability of the proteins, so like if you engineer them to be charged differently, is there a possibility or is there a way to test for if there's other like conditions, environmental factors in a human body that would further negatively affect?
Sure, yeah, absolutely. So characterizing the stability of the proteins is part and parcel of what we do. For the charge-engineered enzyme that I showed you, the melting temperature, I believe, was almost exactly the same as the wild-type protein, if I remember correctly. We also assessed how susceptible was wild-type lysozyme and the engineered lysozyme to degradation by proteases. Those are other proteins that are present in the infected lung, so they chew up proteins. We were afraid the engineered enzyme might be more susceptible to proteolytic degradation. It wasn't. It was exactly the same as wild-type lysozyme. So to the extent that we can, we test that in the lab. And then I think that the in vivo model data is another great case of how do you test for these unseen possibilities, right? Where the engineered molecule may be compromised by something we didn't anticipate. And the data that we have so far says that the engineered enzyme I showed you today is actually good.